In this chapter, we're going to learn about some application field with the protein. There are two terms are actually popular: protein engineering and protein design. Protein engineering may represent the approaches by which you can change some residues of pre-existing proteins to get altered proteins in functions and structure. Protein design is to get ton of proteins. Knowledge of protein tertiary structure is a prerequisite for the proper understanding and engineering of its functions and structure. So we may start the prediction of secondary and tertiary structure of some simple cases that will be homologous proteins. What is a homologous proteins? Basically, those proteins may evolve from a common ancestor gene. Practically, homologous proteins have similar functions and structures. There are some exceptions to this rule. However, in general, this principle can be applied. How can we figure out two proteins are homologous or not? The simplest way is to compare the residue sequences. When two proteins share more than 20, those could be considered as homologous proteins. You can see the relationship between divergence of amino acid sequence and three-dimensional structure of core region of homologous proteins. The difference in main chain atoms of the core regions is plotted against as a function of amino acid homology in x-axis. The curve represents this one, the best fit of the dot real data to an exponential function. So the greater the sequence identity, the more closely related are the scaffold structures. This has important implications for model building of homologous proteins. The more distantly related two proteins are, the more the scaffold must be adjusted to model the new structure. When a novel gene or protein is found, how can we predict the three-dimensional structure of such a new protein based on its residue sequences? First, compare this novel protein with a database of known protein sequences from protein data bank. Next step is to predict its structure based on the structure of known homologous protein. Homologous proteins always contain a core region where the general fold of the polypeptide chain called scaffold are very similar. This core region contains many the secondary structure elements that build up the interior of the proteins. In other words, the scaffold of homologous proteins have very similar three-dimensional structure. But there is certainly difference in sequences, so you must adjust a little bit to accommodate difference in the arrangement of hydrophobic side chains in the interior of the proteins. That's going to be first thing and major thing. And loop regions that connect the building blocks of a scaffold may vary considerably, both in length and in the structure. Therefore, the major task in this area is to predict the structure of loop regions and side chain conformations. The conformation of loops depends on the number of amino acids in the loop and the types of secondary structural elements that it connects, whether they are alpha-alpha, beta-beta, alpha-beta, beta-alpha, something like that, than on the actual amino acid sequences. To model a protein structure, store main chain loop conformations from data bank are attached to the scaffold modeled to have a structure similar to that of non-homologous protein. Finally, the conformations of the side chains are predicted by energy refinement of the model, which minimizes the free energy of the protein by maximizing the interaction energies of the amino acids. Analysis of structures with high resolution has shown that only a few side chain conformations frequently occur. These are called rotamers. We can see real example for such application. Prediction of conformation of 3 CDR in immunoglobulin G has been done correctly. CDRs are loop region involved in antigen binding. These highly variable loop regions are different from antibody to antibody. The 3 CDRs in upper part are predicted. The amino acid sequences of the loop regions were modeled by comparison with the sequences of loop regions selected from a database of known antibody structures. The 3 CDRs at the bottom were obtained by X-ray crystallographic studies. You can see very similar, almost identical structures except the last one, but it turns out the bottom structure determined by experiment was wrong. and later determine to be similar to this one. Next, how can we predict a novel protein that doesn't have any homologous known proteins in data bank? It's not a simple thing. Once the correct secondary structures are known, we may assemble the tertiary structure by packing the secondary elements against each other. We saw some packing issues in chapter 2. Consequently, secondary structure prediction lies at the heart of the prediction of tertiary structure from the amino acid sequence. However, it turns out that it's not easy because secondary structures may be influenced by the tertiary structure. For example, pentapeptide with identical residue sequence that could form whole different variety of secondary structures in real protein structure, like alpha helix sometimes, beta sheet sometimes, loop sometimes. That is, the secondary structures are dependent on the sequence of that region, but also on its environment in the three-dimensional structure. There are over 20 different prediction methods out there. These are classified into two types: empirical statistical method and stereochemical method. The first one, the empirical statistical methods, use parameters obtained from analysis of known sequences and tertiary structures. These methods are based on the assumption that the local sequence in a short region of the polypeptide chain determines local structures. But as you know, this is not the case. The most famous one is Joe-Fassman prediction method and GOR. Those methods just correct 50% in average, sometimes as high as 80%. Second type is based on stereochemical criteria, such as compactness of form with a tightly packed hydrophobic core and a polar surface. You can see Lim method in this category. The prediction, which is similar to the previous one. However, when these large errors occur at the end of alpha helix and beta peptide sheet,